Hello, once again, welcome to another lecture on biology. So we've been talking about genetics, and in this lecture we're going to continue to think about genetics, and we're going to continue to follow along this story that we saw in the previous lecture. So recall that we're looking at this possibility of Jill being the daughter, the offspring of Donald and Daisy, right? And we had already collected 3 pieces of data, or 3 evidences: the freckles, blood antigen type, and blood Rh antigen. And we saw that in all 3 of those, when we looked at the possible offspring of Donald and Daisy, that it was possible in at least one of the combinations of the genotypes that Jill could be the offspring based on the possibilities that were there. So let's continue to look at this and see what happens with the evidence. So now we're going to look at the gene of color vision. So look at this, these circles that come up, and see if you can see the numbers. Most people can see the first number here, number 25. And a lot of people can also see the remaining 5 numbers, number 6, 56, 45, 29, and 8. If you have a hard time seeing these other numbers or can't see them at all, you potentially could have some sort of color blindness. And this particular color blindness is called red-green color blindness. Red-green color blindness is a characteristic that is carried on the X chromosome, and only on the X chromosome. So it is not found on the Y chromosome. For all of the other characteristics that we've looked at so far, they've all been found on autosomal chromosomes. So where it's a homologous pair, and each one of the chromosomes carries all of the exact same genes. But now we're looking at sex chromosomes, X and Y, where the X chromosomes carries a different set of genes from the Y chromosome. So some really interesting things happen in this type of scenario, genetically speaking. So if we look at the characteristics for a normal male will have an X chromosome with a normal color vision. We'll denote that with capital A. And then the Y chromosome does not have this gene, so there's nothing there. We don't put any letters in the superscript. A colorblind male will have X lowercase a and nothing in the Y. A normal female who is a non-carrier will be X big A, X big A. A normal female that is a carrier, so this is a female that can see those colors, can distinguish between the numbers there, is not red-green colorblind, would be an X big A, X little a, and then a female that is colorblind would need to have on both X chromosomes the recessive color blindness gene here, or allele. So little a on both of the X's. And this is one of the reasons why it's very common to see sex-linked characteristics, sex-linked genes like red-green color blindness, to have much more higher frequencies in males and in females, because males only need the one copy in order to be red-green colorblind, whereas females would need both copies to be red-green colorblind. So let's look at how we would code this. If colorblind is colored in, then that means this Donald would be, he's colorblind, so he's X lowercase a y. Jill is colorblind, so she has to be X lowercase a, right? X little a. And we can also then immediately fill in the other genotypes as well, where Daisy needs to be X big A, X little a, because she had to have given at least one little a to Jill, but she can see, so she has to have one that's normal. Jack gets an X with a big A. He didn't get that from Donald, so he had to have gotten it from his mother, but we have no idea what the other X chromosome of the mother would be, so there's a question mark there. So that's how we fill that out. And we put that in our chart across the different things, and then we can ask the question now: if Donald and Daisy would have, if the union of sperm from Donald and from eggs from Daisy came together, what are the possible genotypic outcomes? So we ask our question, what sperm or what kinds of gametes can Donald make? And he can make gametes that are X little a, Y, and Daisy can make X big A, X little a. So you put that in your Punnett square, and here are the possible genotypes. Once again, we can say, well, are one of these genotypes the genotype of Jill? And in fact, it is. So once again, this gene allows for the possibility of Donald being the father of Jill. So let's look at the final characteristic for this pedigree. We're going to look at nail patella disorder. This is located on the 9th chromosome, and some of the characteristics of this disorder, sometimes there are these projections or extensions kind of making a pointy elbow coming off to the side, malformed nails, or these iliac horns, which are these extensions coming off of this part of the hip bone. And if we look in this pedigree, we see that the disorder is present in both Donald and Daisy. This is an autosomal dominant disorder, so if you have at least one copy of this allele, then you are going to present the disorder. So can we fill out the rest of the genotypes? Well, if this is the case, then we know that if you don't have the disorder, then you have to be homozygous recessive. So we can fill in the genotypes for both Jack and Jill and Jack's mother, and we can say, okay, well, we know that they each had to have given one of the recessive forms, but then they present the disease, so they're both heterozygous as well. If we fill out our chart, we can go ahead and put that back down in our chart, and now put our cross up here where we have Donald and Daisy. They're both heterozygotes, big N, little n, big N, little n. What kinds of gametes can Donald produce? A big N and little n gametes, and same thing for Daisy. We put our Punnett square down, and these are the possible genotypes resulting from a Donald and Daisy cross. Well, once again, is that possible in Jill? It is. Jill is little n, and that was one of the possible gametic combinations resulting from a Donald and Daisy cross. So we've now gone through 5 pieces of evidence. All of them do not refute the idea that Donald is the father, so it's possible that Donald is the father. Now, it's important that you ask yourself, though, does this prove that he is the father? And clearly this does not prove he's the father. It's just that every piece of evidence so far does not refute this idea. And remember, this is a really good example of how we do science. We don't think that we've proven this, but we have, in one sense, gathered data to support this hypothesis. However, any number of evidences could come forward and could demonstrate that Donald is not the father. Now, before we finish this discussion up, I want to talk about an interesting phenomenon that also occurs in genetics, and it's called gene linkage. Notice that as we were going along, a lot of times we'd say, this one's found on chromosome 9, and this one's found on the sex chromosome, and so forth. Well, I don't know if you realized, but 2 of the characteristics we talked about, I mentioned, or I showed that they were found on chromosome number 9, blood type and nail patella disorder. When 2 genes are found on the same chromosome, and if they are in close proximity to each other, we consider genes to be linked. And the reason that they are linked is because the alleles, both of the alleles from the same chromosome, are in a sense linked together because they are going to be passed on as a unit into the gametes. There's not going to be independent assortment of the 2 alleles from the same chromosome. Right? You can think about it this way. When we go through meiosis, right, we're going to separate the green chromosome from the orange chromosome. And what types of gametes are going to come from the green chromosome? Well, only I big B, big N. And from the orange chromosome, only little I, little n. For Daisy, the same combination. We're only going to get big I, big B, and big N in eggs, and little I, little n in eggs. So we don't have this possibility of having big I, big B with little n, and little I with big N, because these are found on the same chromosomes, and so there's no way to separate these alleles, right, from each other. They're passed on as a unit along all in the same chromosome. And so the possible genetic combinations then are actually only these 3 possibilities. Big I, big B, big I, big B, big N, big I, big B, little I, big N, little n, and same thing down here, and then little I, little n, little n. Those are the only possible genetic combinations. Now, if though these genes are a little bit apart on the chromosome, then there is a possibility that they could be broken up. You may remember that we talked about gene linkage that can be broken up by crossing over and genetic recombination. So right now what I'm showing is we're at that moment in the cell where the genetic material has been duplicated, and this cell is getting ready to go through the process of meiosis, right, to become eggs and sperm. Recall that at one point during this process, these chromosomes move together and form this tetrad structure, and the tips start to cross over on each other. They form these sites of chiasma, and you get crossing over that occurs, or genetic recombination, where now you have a green chromosome with orange tips, or orange chromosomes with green tips. And when you do that, you have now broken up those linked genes, because now I can get the little I with a big N, and I can get the big I, big B with a little n, different combinations. And it's really interesting because the further that the genes are apart on the chromosome, the more crossing over events occur during meiosis. And so you can actually calculate this, and in the olden days, this was one of the ways they used to map genes across the chromosomes. They would look at how many crossing events occur in the population that they were studying. How many of those occurred? The more that occurred, the further apart the genes were. In fact, once genes kind of get on each end of the chromosome, crossing over events occur about 1.2 times or 1.3 times per chromosome. So you can essentially treat genes that are on each side of the chromosome as independent, because crossing over is always going to break them apart. But when the genes are really close together, we consider them to be linked. And they do have the possibility of becoming unlinked in some cases when genetic recombination or crossing over occurs between the two genes. So that's an important thing to remember as well, that this occurs. And once again, what does this do, crossing over and genetic recombination? Well, it just adds to more of the possible variations that are found in gametes, and it just continues to add to the variation that is needed there for evolution to be fueled and to continue to find those particular variants that happen to be a little bit more successful in the environment.